In Part 2, let's deepen our understanding of Mobius by analyzing sample data using the workflow we created. In this video, we will be using a PC with all modules enabled. You can view the list of licensed modules by opening your key file. This PC was repaired due to a malfunction before its hardware serial number changed at that time. So, let's see what happens if we activate the Mobius by the key file issued for the old serial number. By opening the single pulse analysis workflow from the template folder. Failed. The error window says like the unlicensed module is included. Since the serial number is unique to your PC, reactivate the Mobius again using the new key file that matches it. This time, we could open it properly. Now, let's create a workflow. Open Mobius Editor from the menu bar. The top right area of the window shows the names of multiple modules that can be placed. First, place the replay raw data file. When you click it, the description of it will be showed in the field below. Double-click to place its panel in the empty workflow. Modules that can be connected downstream of it are showed. Double-click, filter raw data, to connect it. Connect one more in the same way. And one more. The arrow next to the module name means the hierarchic relationship. Connect, display all channels, at the end of the stream. Here, let's try removing the third filter raw data. The display all channels lost its input is now highlighted in red. With it selected, double-click a module that can provide an input and connect at the position. The structural problem of the workflow has been resolved. Close the workflow editor for now. Module panels can be positioned anywhere. And you can resize them by clicking and dragging their bottom right corners. Next, let's talk about tab window. From the layout in the top menu, you can add more tab windows for placing modules. Added blank tabs named 2 and 3. You can also rename a tab. With the tab 2 selected, open the workflow editor again. We connected Export Spikes Advanced downstream of the second filter raw data and place its panel on the tab 2. Finally, save the workflow as a name you want. Actually, all the template workflows were created manually in this way as you have watched so far. From here on, let's try analyzing a sample data by using a sample workflow. You can download it from the URL as you look. The hierarchical configuration of the modules in the sample workflow is like this. Associate the file with the extension MOFLO with Mobius so that you can open it by double-clicking. First, select a data file in Replay Raw Data File. If you try to replay with nothing selected, an error window will appear. The analysis workflow generated along with the data file. The data file is loaded in advance. However, moving them to a different disk may eliminate the setting. In that case, select the data file first, confirm the file name is displayed in the filename field, and then click to replay it. The sample data is obtained from 3 to 4 div of DRG culture, applying different dose of capsaicin in each files. In the control, no treatment was applied and the data is like just a baseline noise. In the 0 nanomolar, vehicle was applied, and you will observe the big artifact noise due to it at the beginning. In the 3 nanomolar, you will see the responses at the sum channels immediately after the artifact noise. Here, for your reference, let's look at a demo data from a 16 div, cultured cerebral neural network. As you can see, the network is spontaneously and intensively firing. It is fair to say this difference in firing pattern is a good example of the brain functional specificity. Let's go back on track and analyze the 3 nanomolar data file with detectable firing. First, reduce the artifact due to adding a drug droplet by setting filter raw data modules. We specified a 100 Hz high pass and a 5000 Hz low pass filters. The artifact was attenuated to small spiky components of the sharp rise phase. You don't have to enable both filters. Try turning off the low pass. The baseline noise and action potentials have become slightly bigger. Switch to tab 2 and set the threshold for detecting action potentials as spikes. Here, let's focus on channel 56, where an evident response can be observed. In an ideal data acquisition environment free from unwanted noise, a reasonable threshold for distinguishing between baseline noise and action potentials is often around plus or minus 0.015 millivolts. We detect sharp signals that exceed that amplitude as spikes at a specified time window. The same amplitude thresholds can be applied to all channels by clicking Apply to All from the right-click menu. The detected spikes can be overlaid and displayed in Display Extracted Spikes module. The display range of the Y-scale can be changed by editing the maximum and the minimum values. The display range of the X scale will be changed depending on the time window. Specify any pre-post spike peak amplitude time as the spike duration. These values are the same for all channels and cannot be set individually. Let's move on to tab 3. The spike detection frequency is calculated for the specified time window and displayed as a graph on the time axis. The upper module displays a graph for selected channel. The lower module displays the graph for the total channels. Manually setting thresholds visually by your eyes may introduce bias and be a time-consuming task if there are many channels. We can set thresholds for each channel based on a statistical approach. Mobius divides the data into blocks of 500 milliseconds and processed. Can calculate standard deviations of data points within a block processed at the instant time you clicked. Can set any multiple of that SD as the threshold for each channel. A drawback of this method is that calculating the SD using a block containing many spikes results in a high threshold. If you have data showing no response due to pretreatment or pharmacological inhibition, you can first use those files to set the threshold and then analyze the other files. In this workflow, the modules related to exporting analysis results are placed in two tabs. Save Spikes. In tab 2 exports a list of spike detection times. And Save Spikes Freaks. In tab 3 exports the actual values for each graph as CSV files. Check the Enable box in each module, and enter a text you want to add the file name if necessary. And execute the workflow by clicking the Play and Record button. Finally, click the Stop button to complete the export process. Using these files as input, you can further analyze the spike train in another program, or create graphs in a spreadsheet software.